All right, so we'll move to our next speaker. Yeah, the slides are already there. So I'd like to present you Cas Wognum, who will present the work Polaris, an industry-led initiative to critically assess machine learning for real-world drug discovery. Cas is a machine learning engineer at Valence Labs. Within Valence, he has contributed to several open source projects in the data model ecosystem and the different toolkits, which led to the Polaris project, which he will present today. He holds a master's degree in artificial intelligence from the University of Utrecht in the Netherlands.
Yes. All right. Good morning, everyone, and thank you for the introduction, Tom. So today I'll be presenting, I'm actually very excited to present Polaris, which is an industry-led initiative to critically assess machine learning in drug discovery. And we'll unpack what all of that means in the next 30 minutes or so. But before we do, or to get us started, I would like to give a bit of an introduction as to the challenges we face when we're trying to benchmark machine learning models within drug discovery. So I think we can all agree that benchmarks play a quite important role within machine learning research. That's broadly applicable. That's not just drug discovery. I would say that they don't just provide us with a way to measure performance or to track progress. I would say they actually also inspire entire new research directions or even unlock breakthrough results. I would say that the gold standard within evaluating or within machine learning benchmarks is a competition. What do I mean with a competition? I mean to say that the data that is used to evaluate the model is ideally like newly generated and unknown to the people that are modeling that data. And I think within drug discovery, the goal, like one of the most famous examples is perhaps CASP, which is this competition for predicting the protein structure. I think most well known because it was AlphaFold that submitted their model, their predictions to that specific competitions. However, one thing I think is worth highlighting is that CASP wasn't a one-time event. It's actually I think it's a lot of the value comes from the organizers quite rigorously, difficult word, trying to identify the most valuable innovations within the community with a certain like regular interval. Yes. Okay, so we know how to do benchmarking really, really well, right? We can do these competitions. So why don't we just do more of that? And the answer is unfortunately quite simple. It's just data generation is expensive. So I'm putting some numbers on the slides with certain assays, how much it costs to test for a single molecule. And you'll just quickly see that it's not feasible to run competitions with new data with a certain interval, especially given how high the pace of innovation is within the machine learning community. So we need something else than competitions. And what can that be? That's where a static benchmark comes into the picture. So in a static benchmark, different from a competition, the test set is known. You're taking some data that is publicly available, you split it in a train and test set, and then you train on the train set, evaluate on the test set. Unfortunately, there's some issues with static benchmarks. Results, like the evaluation on a static benchmark, given that the test set is known, is more likely to be biased. This can be due to test set leakage, but it's also just because the incentive is there to do really well on this test set. So even sort of like without bad intentions, you might find yourself training like 100 different models, evaluating them all on the same test set and just picking the one that does best. But the question is that still like a valuable innovation at that point? This is a problem, I think, for all of machine learning. This is not just drug discovery, and I think a lot of us are familiar with this challenge. I think one additional point that's worth making is that these challenges are further amplified within drug discovery. Within drug discovery, I think the biggest like differentiator, or within ML for science, perhaps more broadly, is that the data that we're working with is no longer intuitive, right? If I train a computer vision model, I can look at some of its predictions and I can do a pretty good job without any formal training, simply because I'm human, to understand whether that model is doing well or not. I cannot do this within drug discovery. I think a lot of people, like chemistry, biology, physics, these are all fields that people do their entire PhDs on, and they just understand a small portion of it. And the data is just less intuitive to work with. There's other factors, such as that the data is more variable, which we'll get into in a bit. But I just want to make the point that I think these challenges are further amplified within drug discovery. Okay, so what sort of situation is this creating? So I'm actually a computer scientist by training. I'm not a biologist or a chemist. I actually think I wish I would have done more of that in high school, but I didn't. So what happened when I first joined and first started applying my machine learning skills to drug discovery? Well, I did everything I was taught to do, right? I selected a data set, I split my data set. I actually tried to make very informed choices as to how to split the data set. I did hyperparameter search and all of the above. And at the end of that sort of trajectory, I had a 1% improvement over the state of the art. Hooray, we solved drug discovery, right? No, of course not. It's unfortunately not that easy, because then you bring those results to your colleagues who have a more of a background in chemistry or biology or drug discovery in general, and they'll start asking you all these like annoying questions. What's the statistical significance of the results, right? What's the applicability domain of your model? Those compounds you generated, they look great, but there's no way we can synthesize them. How did you handle stereochemistry during dataset curation? Could you actually explain the predictions to me? I just want to make sure we're doing the right thing here. What's the decisional impact? And even questions that maybe as a computer scientist sound simple, turn out to come up quite a lot, right? Like, how do I actually install your model? How do I use your model? So the importance, well, one thing maybe worth saying, like, I don't expect you to know what all of this means, but I think that's precisely the point, right? Like, as a machine learning scientist, you're not trained to think about these things from the get-go, and you might have picked up on it here and there, but you're not really aware of the implications of all these questions. I think given the importance of benchmarks within the machine learning community and pairing that with the inherent difficulty of proposing a benchmark, this is leading to a situation where there is a growing gap between perceived progress and actual real-world impact. And that's something that we set out to address. So everything I've said so far is perhaps a bit on a high level, right? I've tried to convince you that machine learning and drug discovery is difficult. I would like to illustrate that with some examples just to bring the point across. So first things first, if you want to benchmark your machine learning models, you'll need a data set. And data set is, like, I would say in drug discovery, there's quite a bit of data publicly available within databases such as Campbell. But this data is very heterogeneous, it's imbalanced, and it's subject to experimental noise. So to actually curate a data set from such large public databases, you need to very well understand the underlying data generation process and the underlying data modalities. There's this interesting, it was first a blog post by Greg Landrum and then turned into a paper. If you don't know Greg Landrum, he's one of the main contributors or the main maintainer of RDKit. He has a really nice blog post worth reading. But one of the things he did is he did this simple experiment, like, okay, if I just aggregate all the data from these public databases in the most simple way, doing some sort of sanity checks, but not looking into too much of the details, what's the effect of that? And he found that it actually led to quite noisy data sets. So you can see that on the top left. He then came up with a way to improve the automatic data creation by having more stringent like filtering criteria. And he definitely managed to improve this, the situation, as we can see in the bottom, sort of the bottom row. However, one thing that's worth noting in this particular assay, the IC50 assay, the experimental noise of the assay itself is normally estimated about 0.3 delta on this axis here I'm showing. So we can see that even though with this automated curation, he managed to sort of reduce the variance between replicates, he's still above the experimental noise. So we can still expect there to be some noise in this data set. Okay, so what do you need to do if you want to improve things even further, or maybe what should you do nonetheless? And there was this other interesting figure within our space, Pat Walters, and he also writes a great blog. If you haven't heard, I highly recommend it. And he actually wrote a quite opinionated blog post on some of the things you should do to sort of further curate down your data set and make sure you have a high quality data set. Okay, so that's the data. Then the next question is, okay, now we have this data, how do we turn that into a machine learning task, and how do we evaluate performance on that machine learning task? So that is the questions such as, how do I split my data set, or what metrics should I use to evaluate performance? And here again, you need you need a good understanding of the downstream application you're trying to model in order to make the right choices and how to split your data and how to evaluate which metrics to use to evaluate performance, right? So if we think about splitting, I think a lot of people in the room will know that it's probably a good idea to use a scaffold split rather than a random split, because that's a more difficult splitting method. However, it's actually not that straightforward, right? Like if you use the Mirko scaffold, there was another nice blog post by Greg Landrum again, and he showed if you use the Mirko scaffold, that doesn't really align with the intuition that humans have as to what a scaffold is. So what you end up doing is actually you find that there's very few molecules per scaffold if you do it in this automated way, and it ends up being a little bit more like a random split rather than a completely like scaffold split. Now we can even go one step further and ask ourselves, okay, we know that the scaffold split is more difficult than the random split, but is it difficult enough? Or is it perhaps too difficult? And here it really comes down to, again, your downstream application. We wrote this paper at Valence on molecular out of distribution, where we tried addressing this challenge. I don't think we found the perfect answer, but if you're interested in sort of learning about some of the considerations there, I recommend giving it a look. So all of this to say, like, also, like, even if you have a high quality data set, formulating a task that is representative of real world drug discovery remains a challenge. Okay, and the final thing I want to just quickly bring up is the difficulty of method comparison. So now that we have our performance metrics, how do we actually tell whether one method is better than the other? And what we're used to seeing is these, or maybe that's a bit of an oversimplification, but one of the ways in people can present the result is with these tables, right? These leaderboard-like tables, where in bold you get the method that did best. I think one thing that's worth highlighting here is that machine learning model, or like any data-driven model really, is subject to some forms of uncertainty, right? I think an interesting paper there is torch.manual seed 3407 is all you need. This just showed that if you just change the seed of your model before you start training, you can actually see quite a big variance of performance in the final results. Now, besides introducing uncertainty in the model architecture itself through the seed, there's also a lot of uncertainty that's coming from the data, right? If I do a specific split, and then like if I do cross-fault validation and I train my model on multiple splits, you'll see that the results are not equal across all these different splits. Actually, even more so, because drug discovery, the data is so heterogeneous and imbalanced and noisy, you'll find that the variability is even larger. So you should be using some sort of way to sample performance for different splits and different seeds. Cross-validation is a good option here. You should also be using appropriate statistical tests to then compare those distributions. But again, I think this just nowadays isn't really, well, I'm not sure. I'm curious to hear your thoughts afterwards, but like this nowadays is not really being thought anymore within the typical machine learning paradigm, because if we're comparing it to computer vision, data sets there are so large that any result can be assumed to be statistically significant. But here within drug discovery, this becomes all the more important again. And actually, I think within computer vision, it's important too, but that's another topic. Okay, so those are some of the challenges that we observed. So then we thought, how could we go about addressing some of these challenges, or at least making an effort to? And what we came up with was Polaris. So Polaris is a industry-led initiative, and the goal of this initiative is to close the gap between perceived performance and actual impact. I think it's worth highlighting that what we're not doing is we're not proposing one specific benchmark or a set of benchmarks that we have carefully curated and that we've tried to popularize among the community. I think what we're rather trying to do is we're trying to lay the foundation where other people, where the entire community can then start building upon to work with more high quality data sets, work with more high quality results, high quality benchmarks. So specifically, what does that mean? We're working on three things. First and foremost, we're working on a recommended set of benchmarks and data sets. So of all the, this can mean two things. So like of all the benchmarks and data sets that have already been proposed within our community, which ones of these are of high quality and should you be using? But this can also mean perhaps that there is a need for additional data generation. Perhaps we don't have the data to solve a particular problem, and then that's something we're interested in exploring as well. Second is that we want to have guidelines for dataset curation, model evaluation, and model comparison. Here, the goal is really to make these guidelines very beginner friendly, so that independent of your background, you can just read these papers and you have a good understanding of some of the best practices, such that it's easier to do the right thing. And finally, we're also working on open source software. So the thinking here was, we can write the most fantastic paper and publish it in Nature or whatnot. But we were worried that if we didn't actually follow up with it, make it easy to implement these guidelines, that people would not be inclined to use them. So we want to make sure that there's software to simplify the adoption. I think it's finally, I think it's worth saying that we have this grand vision of doing this for all of drug discovery, of all of machine learning based drug discovery. That's a big challenge, just to get started and to sort of see the format that we can use to structure this collaboration. We're focusing initially on small molecule predictive modeling. This is not something we're doing alone. I don't think we have all the answers. I don't think any company has all the answers. So that's why we made a very specific effort to work together with some of the key opinion leaders within our field. And you can see they're all from different companies as well. I think that's important. And one final thing I wanna just highlight is this beautiful quote: "Don't let the perfect be the enemy of the good." I don't think we will have all the answers. We'll do our best to come up with sort of like a good first pass. But ultimately, I think this really needs to be a community-run initiative. We need to sort of, everyone needs to chime in and share their opinions. So I don't think we will be in a position where on day one, we'll have solved this problem. What we're rather trying to do is just work towards, like, just make some progress, try to improve the state of the art. And I think there's some low-hanging fruits that we can already address right now. Okay. Then finally, I want to just quickly touch upon what we have been calling the hub. So if you remember this third bullet, we are working on this open source software to simplify the adoption of these guidelines that we come up with. And that open source software will be centered around the hub. So what's the hub? The hub is a benchmarking platform. So think of like Kaggle or Hugging Face. However, we've built it from scratch specifically to address some of the challenges that we're facing in drug discovery. The mission is to make it easy to do the right thing and wrong to do the hard thing, right? So if you're using this platform, whatever guidelines or standards we come up with, we want to make it as easy as possible to implement those standards in your research by default. In the current state, so that's the mission, that's where we're working towards, but this is like an ongoing process. In its current state, I would say it best described as like a source of truth. So we've built out this platform to host the wide variety of data sets and benchmarks you might encounter in drug discovery. And if you use our platform, you'll be absolutely sure that the data set you're using like this week will be the same, it will be the exact same data set as if you access that same data set in another month from now. Like we want to make sure that there's sort of that we're operating as a source of truth. So this platform is built to host a wide variety of data sets in drug discovery. So whether you're working in phenomics, or you're working with proteins, or you're working with conformers, or you're working with just 2D molecules, or anything else you can come up with, we aim to support it. The way we're thinking about this is, all these different modalities have their dedicated file format. So we were like, there's a lot of complexity in supporting all of that. However, we're really building this platform to be a machine learning ready platform. So what we're thinking is, independent of whatever modality you're using, at the end of it, you'll have to convert it to some sort of ND array as input to your machine learning model. So our platform uses some cool technology to host and the arrays, essentially. We're also making we're doing our very best to make the barrier of entry as low as possible. So if you have data that is currently saved in an SDF file, we can automatically convert it to something that's compatible within Polaris. And finally, no, not finally, but next, we are also we're supporting the wide variety of dataset sizes that you encounter in drug discovery as well. So if you're working in small molecule drug discovery, you might be used to only working with a couple of kilobytes, maybe megabytes. However, if you're working in phenomics, datasets can get very large. So there's one that Recursion open sourced, which was about 83 terabytes. And we're aiming to support like everything within that range. And finally, we also put a lot of effort in making sure that out of the box, you have quite performant data access. So whether you use this largest data sets of 83 terabytes and you load the data directly from the cloud, or you're using a small data set, we made a lot of optimizations to make sure that sort of the out of the box data loader is fast and can be used within machine learning. Okay, so all of what I've been saying so far is no longer, well, still a prototype, but it's not just in my head, it actually exists. And this is the API that we ended up with. The cool thing is that this API will be the same, independent of what your underlying data set is. So that hopefully makes it also very easy to transfer your skills from one domain to the next. Okay, so what's then next? So the current state is this, where we built this sort of like general purpose platform, which really operates as a source of truth. We're already working on a couple other things. So one thing we're working on is competitions. Right now, whenever we evaluate predictions, we do that on your local machine, which means that we need the client can get access to the test set if they really wanted to. We're working on a scenario where that's no longer needed. So you actually upload your predictions to our server. We'll give you back the results. This is just to make the evaluation less biased, but also to support hosting competitions that people are running within the community. Another thing we're working on is data visualization. So if you upload a data set, we can automatically visualize the molecules that are in it, for example. And another thing we're working on is review or expert review. So, right, like we're coming up with these guidelines. One thing I should have said earlier, but this platform is going to be community run. So anyone here that wants to upload a data set, you can. That does lead to a bit of an interesting case where, okay, if anyone can, how do we make sure that people can still find the resources that are the highest quality? And that's where this expert review sort of comes in. So you can, in a simple format, you can think of this as a comment feature on each data set where people can leave comments saying like, hey, I'm not sure about this, perhaps try changing it, or this seems applicable in this scenario, but not in this scenario. In a more complex scenario, we're thinking of working together with experts within our domain to actually evaluate the data sets and benchmarks, a bit similar to like how you would do that for like peer review within the academic community. And then the question is, what else? And that actually leads me to my final slide. So this platform, like I was saying, it exists, it's live. And if you, we're happy to give everyone here like early access. We just ask you to follow the Twitter account, Polaris HQ. You'll get a link, you can sign in, you can start using it from there on out. And the question really is like, we've built this platform out to sort of like a minimum that we think is needed. And now we're really interested in learning what other features would you like, right? Like what works, what doesn't work, what assumptions we made make sense, what assumptions we made don't make sense. So that's where we're really interested in hearing all of your feedback. And yeah, with that, thank you everyone for listening. And if there's any questions, I'm happy to take them.
Yeah, thanks a lot for the great talk. Like it's really interesting to learn about Polaris, and I can't wait to see the outcome of those big competitions. As a reminder, like you can line up at the mics if you have questions. So we are a bit in advance on schedule. Like don't hesitate. We'll have time for a few more questions. After that, we'll be heading to the poster session. Thank you.
Hi, thanks for the great talk. I wanted to ask, like, is there there's a lack of like relative benchmarking in which we are creating data sets, not just one single data set, but multiple data sets with known signal to target ratios across them, which helps get a lot of the noise sort of similar across each of them. And I was wondering if that's something you knew as to like why we don't do much of that right now. Is that because we are still getting a hang of benchmarking in general?
Yeah, so your question is, let me try repeat it to see if I understand. So the question is, there seems to be a better way in which we could generate data sets for benchmarking, but we're not really using that as of now, right? Yeah, so I think that's a good question. I think we're seeing a shift, but traditionally speaking, most data generated would serve the purpose of running a drug discovery program, right? These programs are really expensive, so we would try to sort of leverage the capacity that we have towards progressing a program rather than training a machine learning model. I think we're seeing a bit of a shift. I think more companies are seeing the value of generating data sets specifically for training machine learning models. There recently was a competition announced on Kaggle by Leash Bio. They have an example where I think that seems to be like very thoughtful of like, this is meant to be a chemistry or like a machine learning for chemistry data set. So yeah, I think that's the reason why we're not seeing a lot of it, but I do think we're seeing a shift. Thank you.
Thank you. A follow up on that question and the fact that of data set releases and competition, is there any hope, in your opinion, to see like competitions where data is provided by a pharma company or biotech company, and then generative models are then tested, like molecules are synthesized in the lab and tested in the assays? Or is that something that we can only dream of?
Yeah, good question. I don't think it's entirely unrealistic. It's definitely ambitious though. And I'm not sure like within what time span we'll see that work out. But like, I think one of the examples where they're at least trying to do something like this seems to be cache, which is another challenge similar to like Cas, but then for hit finding. And there they actually, I'm not sure if they do synthesis, but at least they let you like select molecules that you think are hits and then evaluate them. Does that answer your question?
Yeah, that answers a bit. Because I think like there's always going to be a limitation as long as there's a held out test set, right? There needs to be a lab experiment to really validate the prediction, especially with generative AIs these days. Like it's or even virtual screening. Like we don't want just to do property prediction on a small set of molecules, but ideally either virtual screen or generate. Thank you. We'll go with the next question.
Thanks. That was a great talk and sounds really exciting. I guess I have two questions. So the first one is, do you plan to have support for data set and split versioning on this platform? Because I would say for the first one, people tend to disagree about the quality of assays, stuff like that. So different individual knowledges. And then for the split versioning, people also disagree on the best way to evaluate the models for a particular task. And then the second question is, if Valence were to like no longer be interested in this area, do you think there's a way for the community to basically take everything that's been implemented and kind of mirror it so that it can be preserved, even if the industry interest is kind of diminished?
Yeah, no, that's a great question. And actually, I completely forgot to touch upon that. So this is lovely. In terms of the second question, it's all going to be open source and open science. So, right, we use services for the web platform specifically that requires us to pay some fees. So it's not really easy to open source that. People like we can open source it, but people won't be able to run it locally without the necessary API keys and whatnot. But the intention is really for this to be open source and open science. I can't stress it enough. I think it's really important that this is like community driven and not a single point of failure with one company that sort of like controls all of it. No, it really needs to be open source and open science. For your other question on versioning, yeah, that's a great question as well. So we don't have versioning as in you can upload one data set and there's multiple sub versions. However, what we do have is once you upload a data set, you can no longer change it. So there you can do some sort of like implicit versioning, but just uploading 3 times a data set. And there's no limit on the amount of data sets you can upload and just naming them V1, V2, V3. This is a very deliberate choice. I think we want to make sure that it's a source of truth. So if you try something again in like a year from now, it's the same data set. And the question was like, benchmarks are based on data sets, results are based on benchmarks in the way we've implemented it. So if you change the data set, what sort of happens in a cascade down that line? So we decided to make it just very clear, like once you upload it, you cannot change it anymore. Yeah, does that answer your question?
Yeah, awesome. Thank you.
Cool, thanks.
Hi, thanks again. Great talk. Synthetic chemists working with the group on some analytical methods. To touch on a point there you mentioned about synthetic accessibility and data sets, wondering if some of the data sets that are being introduced have considered the impact of negative reaction data and that impact. I know with your background from computer science, that would be interesting. Wondering if you could just elaborate on the importance or role or absence of negative reaction data for these data sets.
Yeah, so I can answer that, I think, in a pretty straightforward way. So no, we don't have that right now. So there is a focus on like small molecule predictive modeling, and we're working with people to upload some of the data sets ourselves. However, I do want to invite you, like, if you think that's a data set that should be on there, even if you don't have time to do that yourself, point us towards the right resources. You can do this through GitHub or Twitter or whatever you prefer. And we're happy to like look into it and like see what how much of that we can do to kickstart the platform.
Thanks.
Thanks.